Okay, so today we are going to discuss about DNA barcoding, the concept, method, and applications. I am Professor Subrata Trivedi from the Department of Biotechnology, Haldia Institute of Technology. So, what is a DNA barcode? A DNA barcoding is a method of species identification using a short section of DNA from a specific gene or genes. Just like in the supermarket, when we go to buy some items, we see that there is a barcode in the item, which is scanned by the laser beam or the scanner, and we get the detailed information about the item. So here we use the species, any species. It may be plant, animal, human, uh, bacteria, virus, any species. Then we extract the DNA and we amplify the specific region, short standardized region of the DNA, which is referred as barcode. So that barcode can be used for identification and other purposes. It is a very good and uh, molecular technique for species identification. So the concept of DNA barcoding was put forward by Professor Paul Howard in 2003 while serving at University of Guelph, Canada. And the first publication was biological identification through DNA barcodes. So this publication was very important step in the DNA barcoding history. Here, a short standardized genomic region of mitochondrial cytochrome C oxidase subunit 1 gene was proposed as barcode. You know that mitochondrial DNA is circular and it has many advantages. Number one, it is found in high copy number. That means during barcoding, we do not need to isolate the mitochondria to get the mitochondrial DNA. Rather, when we isolate the genomic DNA, due to the high copy number, we also get the mitochondrial DNA. So, no need to isolate mitochondrial DNA separately. We can just isolate the genomic DNA, and when we do the PCR using the specific primers for the cytochrome oxidase subunit 1 gene, then we can get the barcode. The Consortium for Barcode of Life, or CBOL, was established to support worldwide DNA barcoding, and subsequently an international online data management system, the Barcode of Life Data Systems, or BOLD, came into effect. Here you can see the cytochrome oxidase subunit 1 gene in the entire mitochondrial genome. It is the mitochondrial genome is about 16.5 kilobase pairs. That is 16,500 kilobase pairs. And as you can see, there are two strands, the light strand and the heavy strand. And cytochrome C oxidase subunit 1, you can see it is depicted by the yellow color. It is in the outer strand. Other parts of the mitochondrial genome is shown here. For example, the D loop or the displacement loop is also there in the upper part, the blue color. It is sometimes used as breed specific barcoding, and other regions are also shown here. So we will concentrate our discussion on cytochrome C oxidase subunit 1. There are, of course, there are some other barcode regions also. So in this discussion, we will consider this region only. Another advantage of cytochrome mitochondrial DNA is that it is maternally inherited. That means if we can trace the mitochondrial DNA, we can trace the ancestry also. So now let us go to the next slide. This is Professor Paul Howard, who is regarded as the father of DNA barcoding. And now he is the Chief Executive Officer of Center for Biodiversity Genomics, or CBG. The CBG is the global leader in the development of and applications of DNA-based systems for species identification. This is also in Canada. Now, International Barcode of Life, BioScan is now in place, and BioScan is laying the foundation for a global biodiversity monitoring system and to explore its socio-economic implications. It will achieve its scientific goals by coupling a massive strategic sampling campaign with technologies that have never been integrated and implemented at a scale required for the planetary biosurveillance. So BioScan is in place now, and of course DNA barcoding has gained huge momentum, and there are many barcoding separate barcoding initiatives being taken. For example, CMARS for the Center for Marine Barcoding is there. Then we have different barcoding initiatives for fishes, then for Arctic region, and also for different taxa. So why we need to barcode the different species of our planet? This is a big question. Why we need to barcode? Because our planet houses large number of species and a diverse range of flora and fauna, and many of these species are yet to be discovered. So until now, most of the biodiversity assessment has been done by traditional morphological approaches, and identification of species using taxonomic approaches are often burdened with taxonomic controversies. We have seen that different groups have placed the same species under different taxonomic classification. So when we report a species, sometimes and mostly we face problems to identify the species and where to place. So in that case, we can use this modern molecular technique of DNA barcoding to validate the different organisms that dwell in our planet. So let us discuss the main benefits of DNA barcode. one main aim is to discover new species, and in fact lot of species have been discovered by using DNA barcoding technique. I know many new reptiles have been identified through DNA barcoding. And DNA barcoding can also be a very effective tool in assessment of cryptic species. So what is cryptic species? Cryptic species is a species which is molecularly different, but it is morphologically identical. That means cryptic species are those species which are morphologically identical but genetically distinct. So these cryptic species have very high chances to give rise to new species. So identification of cryptic species is very much possible by using this DNA barcoding technique. Now coming to another point, just imagine the marine ecosystem. It is huge ecosystem covering more than 2/3 area of our planet. So DNA barcoding can accurately link the larval stages of the species in order to unravel the life cycle of different marine species. It is very difficult and in many cases quite impossible to link the different life cycle stages of the marine species because the ecosystem is very huge and diverse. So the barcode is same for the egg, larvae or any developmental stage and adult. So we can easily link the different stages to have a clear idea of the life cycle. So coming to the very important point, next point, that is DNA barcoding can be used for rapid identification, authentication and safety assessment of foods, including seafoods, whether it may be cooked food, canned food, smoked products or the products which we get in the restaurants. In fact, one study has shown that in Canada alone, 40% of the fish species that is offered in the restaurant are not the same species which are claimed in the menu chart. So nowadays it is routinely used for seafood and other food identification and authentication. Now another important role of DNA barcoding is that it is used to detect and prevent the trading of endangered species and their body parts. There are many endangered species and they are under IUCN. You can see the red data list. Some are threatened, some are nearly threatened, some are least concerned and some are endangered. So many endangered species are illegally traded. Normally what happens, they cut the body parts into pieces and then they trade it. So sometimes it is very difficult to identify the species when they are cut into different parts, but DNA barcoding can easily identify those species and the proper preventive measures can be taken. Another very important topic related to DNA barcoding is that it can be used for early detection and quick detection of invasive alien species or IAS. What is invasive alien species? These invasive alien species are one of the most important direct drivers of biodiversity loss. You know, now the loss of biodiversity is a huge concern because of the loss of habitat and pollution, climate change, and other uh, anthropogenic stresses. The biodiversity loss is a serious concern. So this IAS may be introduced accidentally or deliberately into natural environment where they are not normally found. So from the name you can understand that invasive alien species are those species that are not normally found in the habitat, but they are introduced from some other place. For example, when the ships go from one international water, or like from Europe to Australia, and the ballast water is released, they contain some species. They may contain invasive alien species. These species, when they are released to the environment, they become dominant over the native species. And what happens is that the species diversity index will severely go down and index of dominance will become high. And the dominant species will be the invasive alien species. So it is estimated that in Europe alone, invasive alien species cause an estimated loss of at least 12 billion euros worth of loss every year. And the main cause for biodiversity loss is the introduction of invasive alien species. These invasive alien species have caused havoc in the fragile ecosystems like in the Great Barrier Reef. Some fishes and some starfishes, they have caused huge economic loss because they have overcome, or they are outnumbered. They have outnumbered the normal natural species inhabiting the Great Barrier Reef. Great Barrier Reef, we know that it is a very serious concern now, not only because of this invasive alien species, but also because of coral bleaching and other effects of anthropogenic stress. So this is an IUCN report shows that among the 395 European native species listed as critically endangered, 110 are endangered because of invasive alien species. So the quick detection of the invasive alien species is very important. So DNA barcoding can be used to quickly and accurately identify the invasive alien species, and prompt preventive measures with subsequent regulatory control can be initiated. And there is just to give one information that there is a separate section for invasive alien species in the barcoding initiative. There are many more benefits of DNA barcoding. Because of the time constraint, I am not going to the details. DNA barcoding and molecular clock. What is molecular clock? The molecular clock is a figurative term for a technique that uses the mutation rate of biomolecules. Here we will discuss about DNA. So DNA barcode data can be used for molecular clock analysis, which can predict the approximate time of origin of the particular species. So this is a very good tool nowadays to predict the approximate time of origin of that particular species. Our team has done DNA barcoding of living fossil, that is horseshoe crab. In Bengali we call Jibanto Jivasho, collected from Sundarbans, UNESCO World Heritage Site. So here we have shown that among those living fossils, that is horseshoe crabs, which species originated first, and then from which species another horseshoe crab has originated, and how the migration pattern, everything has been shown, and it is published in our book DNA Barcoding in Marine Perspective. In that book we have published the findings, and we have shown that how this primitive species have migrated from the Tethys Sea. You know, Tethys Sea, it was in the southern part. Then at that time the Indian peninsula was not there. It migrated from the south to give rise to the Himalayas also. So we could link the plate tectonics with the movement of the horseshoe crabs. Now coming to the method, I have made this chart very simple, as simple as possible. First we have to collect the specimen, whether it is plant, animal or bacteria, whatever. And very important part is vouchering. What is vouchering? Vouchering means we have to keep record of the species. For example, if it is a fish species, we have to keep the fish under proper conditions, and we have to assign a voucher number, and it has to be stored for future reference. Then after that we collect the tissue and do the DNA extraction. For example, if it is from plants, we use CTAB method, or for animals we can use phenol chloroform isoamyl alcohol method. Different methods we can apply, and then we do the PCR amplification of the barcode region. Here I am going to specifically tell about the cytochrome oxidase subunit 1, and after PCR we run the PCR product in the gel, agarose gel, and then after viewing the gel we just extract the PCR band and we do the sequencing. After editing and complete just like we have to check the ORF, everything that are required, and then we submit it to the gene bank. The gene bank will offer the accession number, and without accession number the data cannot be published. So after that we do the bioinformatics analysis, and this is the very simplified method. Without going to the details, I have explained. Now this is a picture from abroad. I did our team under my supervision did the DNA barcoding for the first time from the Red Sea fishes. So you can see the collection of sample. Just 2 gram sample is enough for DNA barcoding purposes. So just to give an idea to the students, specifically I am telling my students, because I take the molecular biology theory and lab here. So just we took the sample and we used test buffer for this extraction. Then we did the standardized phenol chloroform isoamyl alcohol method for DNA isolation. So just to give an idea, I use this forward primer and reverse primer shown in green color and red color, and normal PCR we performed. Just I have mentioned the in red color the annealing temperature. Annealing temperature is very important because if the annealing temperature is not right, then the PCR will not give the desired result. So you can see that we got the PCR amplified fragments about 655 base pair, and the fragments which are less than 500 base pair are not accepted by the board, and so it should be at least 500 base pair. And then we recovered their DNA bands by using PCR purification kit and amplified by using automated DNA sequencer. So then we did the open reading frame, check the open reading frame to detect the correct amino acid sequences and submitted to the NCPI database. And this is the bioinformatics analysis of the sequences obtained. And just a very short description about the 1, 2, 3, 4, 5, 6 fish species which were collected from the Red Sea. And the local name in Arabic is given, and the common name is also there, scientific name, and the accession number. If you put the accession number in the gene bank, NCBI, then you can get the details about each species which we have submitted, and the sequence length of in base pair is also shown there. So the DNA barcoding of Red Sea fishes from Saudi Arabia, this was the first approach. Then the paper came in DNA barcodes. Then we have several publications on DNA barcoding. Just like one of them is the role of DNA barcoding in marine biodiversity assessment and conservation. Then our books on DNA barcoding, both published by Springer International. First one is DNA barcoding in marine perspectives, assessment and conservation of biodiversity. And after the success of the first book, we were requested by Springer to publish the second book, which was DNA barcoding and molecular phylogeny. And lot of important revelations from DNA barcoding has come up in our book. Now, because time constraint is there, so I am not discussing the details, but just I gave a overall view of DNA barcoding, and hope our students and researchers will benefit from this very short video. Thank you very much. See you next time. Thank you.